Hello, everybody. Today, this is continuation from the last lecture or slides about agricultural biotechnology, of course, and uh, continuation from agricultural biotechnology. Therefore, we shall see starting from this part to the last part of this text, Unit One. And one aspect of biotechnology as applied to agriculture is the production of transgenic plants. Transgenic animals, of course. Therefore, transgenic plants have been genetically modified to have these features like disease resistance, herbicide resistance, and pesticide resistance, better nutritional value, um, better shelf life, and also drought tolerance. And many other traits have been given to plants through genetic engineering or genetic modification or by inserting genes from other organisms. Transgenics refers to the transfer of gene from one species into another. Otherwise, if we are transferring genes from between two organisms of the same species, that is another version of genetic engineering, which is known as cisgenics. Cisgenics. In this case, we are transferring genes from one organism, one species into another species, between two different species, that is transgenics. Therefore, the most common type of gene transfer is, of course, transgenics, not cisgenics. Cisgenics may be used in cases like to transfer normal genes in place of abnormal genes in gene therapy. During this time, of course, you give genes genes of genes of one organism into another, both of which belong to the same species, of course. Otherwise, in transgenics, plants or animals are given genes from another species. Now, in agricultural biotechnology, one production of transgenic plants is concerned with giving genes for plants to make them disease resistant, to make them herbicide resistant or tolerant, to make them pesticide resistant, to have better nutritional value, better shelf life, drought tolerance, etc. And also plants are being engineered. If you further, scientists have moved plant engineering further. Once they further, they can plants are being engineered to produce pharmaceuticals or drugs, or even if you like, we also produce vaccines, which are commonly known as edible vaccines. We can genetically engineer plants to produce uh, proteins that serve as vaccines in their fruit, so that when you eat the fruit, you are benefiting your body with vaccine, so that your body will be ready for defense of that particular pathogen. Then such plants that have received recombinant DNA or genetically transferred DNA from other species are called transgenic plants or genetically modified plants or organisms, if you like. And corn, potato, cotton, soybean, tomato are the first group of crop plants to be genetically modified. And the other aspect, for example, pest resistance, one area of transgenics, one area of genetically engineering plants is to give them pest resistance, ability to give them pest resistance. Therefore, one area of biotechnology in plants is giving the plants genes so that they can produce chemicals that can kill pests. Some plants have been genetically engineered to protect against attack by insect pests. The bacterium, imagine, Bacillus thuringiensis, produces a toxin that kills caterpillar. and larvae, caterpillar, and larvae of other insects that are pests to our crop plants. Uh, the gene for the toxin has been successfully introduced into some plant species using a bacterial vector. I will tell you about which bacteria is being used as a vector later on in our lecture, of course. Yes, bacteria can be used. One bacteria, which is known as Agrobacterium tumefaciens, is genetically a genetic engineer bacteria that can insert this plasmid into plant cell. It sends it from cytoplasm to the nucleus, you see. Therefore, scientists are using this bacteria as a vector so that genes can be inserted into plant tissues. The gene for the toxin, that is Bacillus thuringiensis toxin, that is known as cryprotein, has been successfully introduced into some plant species using a bacterial vector. And the engineered plants produce the toxin and showed increased resistance to attack by insect pests. Imagine you can give the gene for that toxin production for the plants, and the plants will have the ability, genetic ability, to produce that toxin so that they are able to kill insect pests which come to devour them. And example, um, BT cotton and also BT corn. These two are given the gene for cryprotein. Cryprotein is a toxic protein that kills pests from obtained from Bacillus thuringiensis, of course. We give the gene for cryprotein for this one. Therefore, not only this, many other plants have been genetically engineered to be pest resistant. Particular example, corn engineered against its major enemy. The major enemy corn is corn borer, which eats its leaves and then burrows into the stalk, eating its way up until the plant cannot stop. I mean, cannot support the air. Imagine it destroys the plant. You can see this is the worm, the adult mouse, and its larvae here. How it destroys corn, especially number one enemy of the corn plantation. Of course, it destroys the seeds, even it bores into the stem. Therefore, if you engineer this corn plant by taking gene from Bacillus thuringiensis to produce cryprotein, this worm will be killed, or the larva will be killed. And the other is cotton has been engineered to protect it from the boll weevil. The boll weevil. This is, of course, this is tobacco bedworm. The boll weevil, you can see in the diagram. This is one enemy of cotton, of course. So if we genetically engineer cotton by using the Bacillus thuringiensis gene, the cryprotein producing gene, we can control this enemy of the cotton. The other insect resistant tobacco also exists, engineered against tobacco bedworm. To the left you can see tobacco bedworm, the mouse, and the larvae, the worm, the larvae. Therefore these are destroyers. Tobacco is one big industry, though it is not nutritional plant. It's a drug, of course, yeah, but still many economies depend on tobacco for export also. The use of TI now, after we have briefly seen transgenic plants. Of course, transgenic plants is not only giving pest resistance. We can give genes for disease resistance. We can give genes for herbicide resistance. We shall come to that point. Now, earlier I have told you about how one bacterium can naturally perform genetic engineering, able to insert its plasmid into plant tissue. The use of TI plasmid as a vector. This talks about that particular bacterium. We shall see genetic engineering of plants poses problem for biologists. One big problem is genetically engineering plants. How is it a problem? Of course, difficult to insert genes into plant tissues. It is not, the plant cells do not accept plasmids as bacteria cells accept plasmid. This makes genetically engineering plants difficult, as plants do not accept plasmids in the same way as bacteria cells do. One problem. But uh, however, scientists discovered that one particular bacterium called Agrobacterium tumefaciens, which is parasitic to some plants, uh, can perform naturally this genetic engineering. What is this bacterium? Let us see. When the bacterium infects, of course, it's pathogenic to plants. When the bacterium infects the host plant, the specific host plant cells, the bacterium injects its plasmid, now known as TI plasmid. We call it TI small. TI stands for tumor inducing. I will show you. I do have the diagram. I will show you immediately. This plasmid has genes, and when these genes are in the plasmid, when the plasmid is given to the plant, it goes to the nucleus. It is expressed so that it causes that particular group of cells undergo cancer-like development. It multiplies without control and produces a swollen outgrowth in that area, usually the junction between the root and the stem. Therefore, this plasmid, the TI plasmid, goes to the nucleus, into the nucleus of the host plant, where the plasmid transfers the genes for tumor formation into the host plant gene DNA, so that there it expressed and causes what is known as crown gall disease, a cancer-like development. This causes a cancerous growth in the plant called crown gall. I have this diagram. Crown gall resembles just like this. You see the dark area there, perfectly at the junction between the root system and the shoot system. Crown gall. If you like another diagram, you see this is the most common area of infection. The bacterium is soil bacterium. It penetrates the plant. Then it introduces its plasmid into plant cells. The plasmid goes to the nucleus. From the nucleus, the genes are given to the plant DNA, inserted into the plant DNA, and the plant cells develop or grow just without control in that area and forming tumor-like swelling, which is known as crown gall. This is of course abnormality. Therefore, when scientists study how this bacterium causes this swelling, they learned that it is a perfect genetic engineering bacterium. It can send its plasmid and the nucleus. When the plasmid reaches to the nucleus, it inserts its genes for crown gall development into the plant DNA, so it is expressed from there, and the plant will have such character, that is such disease symptom, swelling, cancer-like swelling, you see. And this means that the bacterium acted as a vector to carry genes into plant cells, and that Agrobacterium tumefaciens performs a natural genetic engineering. You can call this bacterium a natural genetic engineering bacterium. A bacterium which can perform genetic engineering on its own. So this was the problem. How can we transfer genes into plant tissue? It was difficult. Thanks for this bacterium. As far as plants susceptible to this bacterium are concerned, you can use this bacterium for you to insert genes by putting the genes that you require, you need to transfer on the plasmid of this bacterium, you see. Therefore, and geneticists then began to use the bacterium as a vector to insert the desired genes into susceptible plants. And first, they take out the tumor-forming harmful gene from the plasmid of the bacterium. Yes, you have to take, because you don't want your plant, your crop plant, to produce crown gall. Just remove that is the disease gene of the bacterium on its plasmid. Then insert the gene they want, or you want, into the plasmid of the bacterium. For example, you can insert the gene for cryprotein synthesis from Bacillus thuringiensis, so that the plant will produce a pesticide, the cryprotein. so that it becomes pest resistant, and then make the bacterium infect plant so that it transfers the desired gene into the plant. Simple and very cheap way of transferring gene by using this bacterium. This shows you while doing, you take sample of cells from a plant, tissue culture, you do tissue culture. During that tissue culture, you use the bacterium to transfer you the gene that you require. Tissue culture, gall formation, finally each gall, broken piece of mass of cells, differentiate as plants are totipotent. You know, I have taught you about this, plant tissue culturing. Just you take sample cell from plants and you provide nutrition, petri dish or flask, and you also use agrobacterium tumefaciens to infect the plant cells so that it will transfer the gene that you want to transfer into the plant. First, the plants divide mass of cells callus. When you break down those callus cells which have received the gene from the bacterium plasmid, into break it, divide it, break it into pieces and transfer it into a fresh medium or media, then each will grow into plantlet. This plantlet now that you get, the small plant that you get, have received genes from the plasmid of the agrobacterium tumefaciens. So you can plant these plants. You can take it to the greenhouse or field and grow with new traits now. For example, giving herbicide resistance to crop plants. Now, let us see how scientists were able to give herbicide resistance. Herbicides are chemicals which kill herbs or weeds, weed killers. So, when we are applying herbicides in our agricultural lands, we don't want to kill our crop plants. Rather, we kill the unwanted plants known as weeds. So, in order to make our crop plants resistant to the herbicide, so that the herbicide only kills the weeds, we have to give herbicide resistance to our crop plants. Herbicides are chemicals made artificially to kill weeds in agricultural field. And one such chemical herbicide which is safe and effective is glyphosate. By the way, glyphosate is one potent herbicide, but one weak point. The problem of glyphosate is that it also kills the crop plants, not only the weeds. You see, it is very powerful to kill weeds, not only weeds, but it can damage your crop plants. You don't want your crop plants to be destroyed. So, to solve this problem, scientists inserted a gene for an enzyme that breaks down glyphosate, along with antibiotic resistance gene into TI plasmid. Imagine the same bacterium is being used to transfer the gene TI plasmid of agrobacterium tumefaciens. Then the genetically modified plasmid reinserted into the bacterium. You put in the bacterium. Then you make yes, therefore, plant cells are then exposed to this transgenic bacterium and cultured on a plate containing antibiotics. The plate contains antibiotics. You have inserted the gene for glyphosate resistance plus the gene for antibiotic resistance. So the medium contains antibiotics. So when you expose these plants to antibiotic containing plate or medium, the plants which have taken the plasmid, the genes, the glyphosate breaking enzyme gene, also take the antibiotic resistance gene. Whereas those plant cells which didn't get the gene will be killed by the antibiotic, so that you have isolated those cells that have taken the gene that you need, that is glyphosate enzyme that breaks down glyphosate. Of course, the only plant cells that grow are those in such a medium, the medium containing antibiotics. The only plant cells that grow are those that have taken up the plasmid, whereas the other cells are killed by the antibiotics. You see, and this is a technique. Additionally, glyphosate is the resistance gene, plasmid. You have you cut tissue culturing, tissue. You cut a piece of plant cells from leaves, and these isolated cells are exposed to the plasmid that contains uh, enzyme that breaks down glyphosate as well as antibiotic resistance gene, and you are putting this into a medium that contains antibiotics. When these cells multiply because there is nutrient, so that the only cells that grow and develop healthy are those which have received the gene, the plasmid that carries glyphosate breaking down enzyme, and the enzyme, I mean the gene that enables the plant cells to be resistant to antibiotics, so that you can multiply this into callus. Callus breaks down into pieces to give rise to seedlings. Of course, it is one fast mechanism of plant propagation, micropropagation, tissue culture. Therefore, we do this under controlled condition in the lab. Let's see now detrimental effects of insect-resistant crops. Let's go back. Insect-resistant crops, as well as of course herbicide-resistant crops, insect-resistant crops, and herbicide-resistant crops. What are detrimental effects? What are the negative aspects of making our crop plants resistant to pests, resistant to herbicides, herbicide tolerance? Of course, while genetically modifying plants would significantly increase crop productivity, there are also fears and risks associated with them. Many cause the insect to develop resistance. You see, this may result continued exposure after a certain period of time. The insects, this may push the insects to undergo change and develop resistance to our pest, I mean to our pesticides. The other is damaging effect on other beneficial insects. We are meant to kill those pests, but if if beneficial insects come, it may also kill non-selective. The gene may be transferred into other unwanted plant species, like you are giving crop plants herbicide resistance, insect resistance. If these crop plants can breed with weeds, for example, so that, which means the gene that you put in crop plants will be transferred into unwanted plants, unintended target, the weeds, so that the weeds are also going to be herbicide resistance and pest resistance, which is the effect that we don't want to bring, and therefore these are the risks associated with genetically modifying plants to make them pest resistant as well as herbicide resistant. Okay, now transgenic animals, from transgenic plants to transgenic animals. A little about transgenic animals. Although several recombinant proteins are successfully produced by genetically engineering bacteria, some genes require eukaryotic animal hosts for proper expression and processing. There are some complex proteins which are difficult to be expressed. The genes are not able to be expressed in prokaryotic cells. Therefore, impossible to express these genes into the transfer, bring those genes into bacterium so that expressed and the gene product is produced. They are not possible to be expressed in prokaryotic cells. They require eukaryotic cell to be expressed and processed fully. Because of this, the desired genes are cloned. You multiply the genes and transfer it into animals such as, as far as history is concerned, animals such as goat, sheep, chicken, mice, so that it could be expressed. For example, in the milk of goat or egg of chicken, and some part of the mice, like liver of mice or brain of mice, whatever. Such animals that are genetically modified are known as transgenic animals, you see. And some human proteins are expressed in the milk of transgenic sheep. goat, and some are expressed in the eggs of chicken, as I've tried to tell you earlier. Mice have been used extensively for expressing and studying the effects of recombinant genes and mutations. Of course, this is especially to study mutation and to study the effects of recombinant genes. Mice are research-based genetic modification has been done on mice. Transgenic animals are organisms whose genomes have been deliberately modified by the introduction of foreign genes, allowing them to express new traits and pass this modification to future generation. Once you change their genes, when they multiply, they pass those genes to the future generation, of course. And application of this plant transgenics. Transgenic animals have a wide range of applications, including biomedical research, after especially the rat or mice. Rat or mice are being genetically modified for this research purpose. Uh, Biomedical research, for example, transgenic mice are often used to study cancer and genetic disorders. Even the effects of genetic modification can be studied by genetically modifying mice. And the other area of application is agriculture. Agriculture. Transgenic livestock or animals can be engineered for improved traits such as disease resistance or enhanced production of milk or meat. For instance, some goats have been modified to produce proteins in their milk that are used in human therapy. The protein expressed in the milk will be extracted and used for human disease treatment. You see, by genetically engineering animals like domestic animals like goats. The other is pharmaceutical production. You can genetically engineer animals, give them genes so that they produce pharmaceutical products. Certain transgenic animals can produce valuable proteins or hormones in their milk or eggs, which can be harvested for medical use. That's it. Examples of transgenic animals. Transgenic mice, the first transgenic animal, of course, mice are the first transgenic animals used extensively in research to study gene function and disease mechanisms. You see, genetic studies are done by genetically modifying mice, of course. Also, you conduct genetic therapy also is tried on mice after genetically modifying them. The other is transgenic goats engineered to produce proteins in their milk that can be used for therapeutic purposes. The other is transgenic fish like salmon fish are the genetically modified salmon that grow faster than their non-modified counterparts. You see, you give genes to the salmon so that the salmon grows twice or even above more than twice the rate of the normal, the non-genetically non-engineered counterpart. Faster growth within short period of time, it becomes big, high amount of protein and fat that is good for consumption to fill the bellies of many in need. It's a very delicious, one of the most loved, delicious types of fish around the world, most like around the world, salmon. Ethical considerations. The creation and use of transgenic animals raises ethical questions regarding animal welfare, environmental impact, and the implication of genetic modification. Of course, one area of concern is the ethical considerations. Some ethical institutions argue against the use of transgenic animals. The other ongoing discussion focus on the balance between scientific advancement, ethical responsibility. You see, you do have challenge from the animal rights groups. No, it's not right. It's not ethical to undergo genetic modification on animals, and therefore they argue. Therefore, you have to break this barrier. You have to abide by this. This is one area of challenge for genetic transgenic animals. Genetically modified organisms are closely monitored by government agencies to ensure that they are fit for human consumption. do not endanger other plants because foreign genes could go into other unintended species in the environment, as I have told you, especially transgenic plants. When genetically modified plants, the genes may pass to the unwanted other plants like weeds. And therefore, also extensive testing is required to ensure ecological stability. Therefore, of course, genetic engineering or genetic modification of living organisms is in its infancy. Many argue that we haven't sufficiently seen the effect of these genetically modified organisms, what they cause on human health, their effect on human health, long-term effect, plus long-term effect in the ecosystem. Many are against GMOs. There are a group, there are people who argue against, strictly argue against total ban of genetic modification, genetically modified croplands and animals, the use of genetically modified animals and plants. uh, Still, some argue that they benefit the humanity in many areas like agriculture, medicine, drug production, and many areas to solve the human problem in agriculture, medicine, and even the industrial products by genetically engineering. If, of course, you know that the human insulin is produced by genetically engineering E. coli. You take gene from human, the insulin gene, insert it bacteria. This bacteria can easily be grown in the lab under controlled conditions so that you can produce insulin for diabetes mellitus patients. This is one area of application. Still, the knowledge of genetics is helping humanity in many areas. Of course, there may be some fear by some group of people, the fact that it needs GMOs are closely monitored by government agents. Because of these complaints, many governments closely monitor these GMOs in order to ensure they are safe for human consumption or do not cause any environmental disaster. And in summary, transgenic animals represent a significant advancement in biotechnology, offering valuable insights into genetics and providing practical applications in medicine, agriculture. Their development continues to evolve, driven by both scientific innovation and ethical consideration. Of course, these are the the opposite poles. Ethical considerations consider the ethics, then scientific innovation. Then we need innovate science in a way it will not harm the environment, it will not destroy humanity or be a problem in humanity. As far as science technology is not harming humanity, helping humanity, as far as it is helping humanity, and as far as it has less ethical concern, we weigh the ethics with its scientific value to humanity. And we proceed by improving this. And the other aspect of this unit is talks a little about briefly, very briefly about cloning. Cloning. Let us start from the definition. Cloning is a method of producing identical copies of genes, cells, or organisms. You see, you can clone a gene, piece of DNA can be multiplied. You remember PCR. What does PCR do? PCR means multiply. You give it sample DNA, it makes it many by amplifying it. Therefore, it is cloning, gene cloning. When a bacteria is dividing by binary fission, one becomes two, two become four. This is cloning, sexual reproduction, naturally occurring. Where when you multiply genes under control condition in a thermal cycler, it is artificial. In vitro cloning, you are cloning gene. And also organisms. Bacteria is organism or a cell, yeah. Therefore, cloning is a method of producing identical copies of genes, cells, or organisms. The products of cloning are called clone. And a clone is a group of genetically identical organisms or cells produced either by asexual reproduction or artificially by cloning techniques. For example, what are identical twins? They are clones. Same genetic material, same exactly. They are clones. They are obtained from zygote or embryo, the result of one sperm fertilizing one egg, giving rise to embryo. This embryo splits into two. These are clones. Identical twins are clones, perfect clones. The main advantage of this technique is that they can make large numbers of plants or animals which are exact copies of a parent with desirable characters. Therefore, one advantage, of course, it is asexual reproduction. And one advantage of asexual reproduction is that you need one special trait, one special quality in an animal or a plant. You don't want that special quality to be lost by hybridization, by mixing genes with other other groups that have different traits. In order to keep homogeneity, the same trait from generation to generation, sexual reproduction, asexually multiplying, or by cloning, you maintain 100% same genetic profile so that all the traits that you get, all the organisms that are produced by cloning or asexual means are of the same character, desirable character. If you like certain animal with best meat quality, and if you clone it, all the clones will have best meat quality or best milk production quality. Therefore, this is the purpose of the benefit of asexual reproduction. You can get the desired character in all the clones that are produced from a single organism by cloning process. Sometimes cloning is used to produce skin or other tissues needed to uh, treat patients in hospitals. You can take stem cells. Stem cells are cloned to made to produce, and you can take stem cells from the skin. You can make them reproduce, stimulate them to reproduce. They will give you the organ skin. You can use this skin for skin transplant in case of burning, when somebody got burned electrical burn or fire burn and lost um, its skin in some part of its body. You can take stem cells from his own healthy part of skin and make skin tissue or the skin organ for transplanting. This is one area of cloning. You can clone cells. Animal cloning, which animals can't be clones as we do in plants. Animals can't be cloned as we do cloning in plants. You see, you remember cloning in plants is the technique we have seen that tissue culture. Tissue culture means you take sample of cells from leaf, stem or root and provide nutrition, suitable environmental condition in the lab, and they divide by mitosis, then give rise to mass of cells which give rise to plantlets. You see, this is simply plant tissue culture is plant cloning. When it comes to animals, animal cloning is difficult, it's impossible. Natural setting, just like the way we cloned plants by tissue culture, we can't we can take any cell from any part of an animal, and that will not give us the whole animal. The reason is that plant cells do have the inherent ability called totipotency, ability of any plant cell to give rise to the whole adult plant. We don't have that power. We animals do not have, our cells do not have that totipotency. The only group of cells that possess this ability in animals are our cells, embryonic stem cells during the embryonic stage. Embryonic stem cells. If you take from the early embryo, some cells, these some cells are a single cell, that cell can give you the whole animal, the whole human or the whole sheep or whatever. And if the early embryo stem cells are divided into pieces, each piece can give rise to the whole organism. Therefore, totipotency is there in animal tissue when we are during the early embryonic stage. Otherwise, we don't have this totipotent just like the plant possess. However, scientists have successfully cloned animals in the lab by using techniques of genetics. You see, naturally it's possible, just like the way we did with plant tissue culture. It's possible, but scientists have done some genetic techniques and were able to take some cell from any part of the body and genetic by genetic techniques and provided nutrition, then gave the plant the cell divided and mass of cells finally grown into the full organism. The first successful reproductive cloning of an adult with known characteristics is Dolly the sheep, the mammals, Dolly the sheep. The base of cloning in this case is the nucleus transplantation technology or nuclear transfer technology. How the scientists did this is depicted in diagram. I'll show you. A nucleus from a cell is extracted generally from an embryonic, not differentiated cell. A nucleus from a cell is extracted generally from an embryonic non-differentiated cell. This nucleus is inserted into a previously enucleated egg cell. Enucleated means its nucleus has been removed. It is egg cell. We need the cytoplasm because the cytoplasm of egg cell contains a food reserve. We need this dividing cell, the nucleus dividing cell, to use this nutrient so that we remove the nucleus from a cell. Then we remove a nucleus from egg cell, and then an electric current is used to fuse the nucleus in the cell and trigger the egg to begin developing. You see, we insert the nucleus that we have removed from the cell into the egg cell that has from which the nucleus has been removed. We are replacing the nucleus. The reconstructed eggs are now reconstructed eggs. We are taking out the nucleus of this egg and we are inserting cell from adult body cells, the nucleus with 46 in case of the sheep, of course, the full set of chromosomes. The reconstructed eggs are then implanted into surrogate mother. You treat hormonally the sheep, the surrogate mother, whose womb we need to use for growth, embryonic development. Surrogate mother, the mother that lends its uterus for embryonic development, where the embryo development will take place. If embryonic development occurs, the new organism will have identical genetic makeup to the organism whose nucleus was used in the transplantation. This is a diagrammatic representation. You see from the other cell, taken from a donor sheep, other cell. Then the nucleus was removed from the egg cells. Unfertilized egg cells were taken from another sheep. The sheep below is the egg donor. From this egg, we taken out the nucleus. The sheep at the top, other cell, it's a body cell, other cell taken. From this other cell, we remove the nucleus. And we remove the nucleus, and the nucleus of the other cell is inserted into the cytoplasm of the egg, from which we have removed the egg. So this newly reconstructed cell is electrically, we use electric current to insert the nucleus of the egg cell, the nucleus of the other cell into the cytoplasm of the egg cell, from which the nucleus has been removed. You see, electricity is being used. Then we transfer this cell, or we wait until it divides 2, 3, 2, 4, 8, into the surrogate mother. The sheep that is in the right top is surrogate mother. This is only lending its uterus for development of this embryo into adult. And after a certain period, that is the usual gestation period, the the sheep is born there, the baby sheep you can see. Therefore, the genetics of the baby sheep you observe here is exactly the same to the genetics of the other cell donating sheep left top. You see? Therefore, this little sheep is the clone of the sheep that has donated other cells. You see? This is cloning the technique. Of course, this cloning technology is in its infancy. I hope it will grow and solve many problems as far as agriculture, many other areas. This is now. This is a scientist and the sheep, Dolly. Yeah, welcome to the Roslin Institute research. Dolly is a cloned sheep who had a mother but no father. Her mother. She is 100% exact copy of her mother because everything, every chromosome has been given from her mother other cell. You see, the scientist known as Ian Wilmot did this in 1997, Gregorian calendar, of course. This is about cloning. Now, biological warfare. The last of this textbook, Unit 1, is biological warfare. What does it mean? Uh, Biological warfare, (BW), is the intentional use of infectious agents, bacteria, virus, fungi, protozoa, etc., or their toxins as biological weapons to kill, to harm, or incapacitate humans or his animals or his domestic plants. You see, biological warfare. We are using living organisms like bacteria, virus, fungi, protozoa, pathogenic, potent pathogens, or we may extract their toxins and apply it to kill or to incapacitate or to by any means to harm humans and domestic animals and plants. And biological weapons, you see, biological warfare is the process, the activity. Biological weapons, the weapons are microbes, of course, include any microorganism, bacteria, virus, fungi, or toxins produced by these microbes. In particular, the 1997 Biological Weapons Convention, they call it BWC, bans the development, production, acquisition, transfer, stockpiling, and use of biological weapons. You see, building this agreement, Biological Weapons Convention, signed in 1972, bans the development of these biological weapons, the use of these biological weapons. But we don't know. Many Western communities may apply it in a secret, sly way, which has been banned. You see, there are some countries that are working against international agreement. America is one. Europeans are the other. Europeans, the Western community, is beyond the law. Signed, agreed and signed. Yeah. Therefore, we have to suspect these biological weapons are being developed, being stockpiled. They do have in their laboratories huge store of potent viruses. One day they may release and kill humanity or his domestic plants or animals. Who knows? Who knows? We should suspect those worldwide pandemic diseases like HIV/AIDS, smallpox that has been controlled, of course, many others. Who knows? May be part of this biological warfare. Bioterrorism. Let us talk in relation to biological warfare or biopids. Let us talk about bioterrorism. Bioterrorism is terrorism involving the intentional release or dissemination of biological agents or biological weapons such as bacteria, virus, insect, fungi, and or their toxins, and maybe in a naturally occurring or a human modified form, in which in much the same way as in biological warfare. Yes, terrorism, just you may talk, I will destroy by this. Even you see, even scientists change the genetics of some pathogenic bacteria. Even they can artificially design viruses, artificially make viruses, DNA, RNA viruses that have huge ability to enter into living organisms and cause serious disease which can kill within very short period of time. Potent. They can genetically change bacteria to become very destructive. They can make bacteria drug resistant so that whatever antibiotic you may use, you can't kill those changed bacteria. They are drug resistant. They are made to be drug resistant. They have the knowledge. They have the resource. The Western community have the resource to alter, change the genetics of bacteria, virus, insects, fungi, and even produce the most potent toxins which can destroy large amount of population within short period of time. Effective than using the machine guns or chemical weapons. These are far effective. Therefore, bioterrorism is a terrorism involving the intentional release or dissemination of biological agents. These biological agents as in their natural form or even by changing their nature. How can they change in their nature? They change their genetics to be very potent, very powerful in causing disease and destroying large number of population, are able to spread very fast. They can make them able to spread very fast in a population. The art of bioterrorism can range from a simple hoax to the actual use of these biological weapons. Simply, you may scare people. I will send you this weapon, this virus. I will release. You simply talk and act as scaremonger. That means you release fear by your own media, fear from, or even you actually release the weapons, the biological agents. A number of nations have or are seeking to acquire biological weapons. There are concerns that terrorist groups or individuals may acquire the technology and expertise to use these destructive agents. Why not? We have to feel. Biological agents may be used for an isolated assassination. You can use it as isolated to kill a certain individual, to cause incapacitation or death to thousands. If the environment is contaminated, a long-term threat to the population could be created. You can go somewhere in Africa and contaminate the people so that it will keep on killing year after year the population. Uh, Even I do suspect the Western community may use this to control the rapidly growing population of the uh, African and Asian continents, of course. Qualities of a good bioweapon. This is not included in your textbook, of course. How can they design good bioweapons? I have tried to utter little about this bioweapon qualities. First, the biological agent must be lethal. They must be killers, potent killers. Second, the weapon should be quick acting when used, fast acting. It must enter into your systems very fast and cause symptoms within few hours. Quick acting, and the biological agent should be easily spreading. It must spread easily so that large number of population will be destroyed, and it should be inexpensive to grow in large quantities. Yes, you should produce it in a cheap way. It should be cheap to produce it in the lab. It should be able to survive outside of the body. It should survive outside the body, in your waste, in your sputum, so that it will keep on spreading. These are the qualities, you see. Bioterrorism agents are typically found in nature but could be mutated or altered to increase, I have told you this, to increase their ability to cause disease, make them resistant to current medicines, or to increase their ability to be spread into the environment. You see, this is one area that scientists are working to make these bioterrorism agents or biological weapons much more destructive, to make them much more destructive. Uh, when used as agents of war. And thank you. This is the end of the first unit. Please stay awake, work hard, and there will be plenty to eat. We say. Thank you.